The overall goal of this procedure is to unravel virulent strategies in host pathogen systems by identifying the interaction of a Candidus phytoplasma moly effector protein with host proteins from Malus domestica.
This method can help answer key questions in many different biological research fields and is here used in plant research to elucidate molecular mechanisms underlying the development of apple proliferation disease. The main advantage of this technique is that a single pathogen effector can be screened against thousands of potential interactors, making this method an easily feasible starting point in infection research.
To begin, identify infected trees with apple proliferation-specific symptoms and control trees that are symptom-free. Using clean secateurs, cut root samples from 3 different sites of the root system that have a diameter of 0.5 to 1 centimeter and a length of about 5 centimeters. Place the root samples into adequately labeled plastic bags. Then place the samples in a cold box with chilled thermal packs and store them at 4 degrees Celsius until further processing. Rinse the root samples with water to remove the soil. Then transfer the samples into a sterile petri dish and use a sterilized scalpel to remove the root epidermis and the cortex. With a clean lint-free tissue, wipe the scalpel. Then dip the instrument in 70% ethanol and heat sterilize it over an open flame. Use the scalpel to scratch the phloem. Chop the sample into small pieces